leukemia with heterozygous mutations within the Fanconi pathway. The disease we are researching is T-cell acute lymphoblastic leukemia, or for short T-ALL. T-ALL is a subtype of leukemia characterized by the aggressive proliferation of T lymphoblasts. This disease remains fatal in 25% of children and 60% of adults. And our main goal of the lab is to find improved therapies for high-risk T-ALL. And my part of this lab is to identify and find drugs and concentrations cells with heterozygous mutations within the Fanconi BRCA pathway are hypersensitive to. This all started when targeted exome sequencing on a cohort of 40 pediatric T-ALL patients revealed that 38% of these patients have heterozygous mutations within the Fanconi pathway. Many of these patients also have mutations within the Notch1 gene and the CDKN2A gene, which is pretty common in T-ALL in general. But as you can see, that there's a pretty large percent of patients with heterozygous mutations within the Fanconi pathway. But you may ask, what exactly is the Fanconi pathway? The Fanconi pathway is a DNA repair pathway where it specializes in the repair of interstring crosslinks. An interstring crosslink is a molecule that bonds to both strands of DNA and obstructs replication. Replication is very important to the cell, and this pathway is used to be able to allow the cell to survive and not go through apoptosis or cell suicide. And it all starts with RPA, which then coats the single-stranded DNA. Then after that, it phosphorylates ATR, which is a kinase, and then ATR phosphorylates complex 1. Complex 1 ubiquinates FANCD2. FANCD2 recruits complex 2, and finally the interstring crosslink is repaired through homologous recombination. Homologous recombination is repaired when you use an identical molecule DNA and use as a template, and be able to repair the double-stranded break due to the interstring crosslink. Usually cancers linked to the mutations in the Fanconi BRCA pathway are ovarian cancer, breast cancer, and AML, and also diseases such as Fanconi anemia. Homozygous mutations within the Fanconi BRCA pathway are also related to thymic lymphomas in previous studies, but never has been shown to be caused in or have mutations in T-ALL. As I said before, in our cohort, all patients that have mutations in the Fanconi pathway are heterozygous. And this is important because many cancers involved in the Fanconi BRCA pathway have mutations in both alleles, where they have loss of both copies, while in T-ALL, we only lose one copy. And our hypothesis is that because we're trying to see whether or not, due to the heterozygous mutations in the Fanconi pathway, if it is more sensitive to drugs that induce or inhibit the DNA repair pathways. And we hypothesize this because due to these heterozygous mutations, the Fanconi pathway is haploinsufficient, which means that this pathway is impaired. And due to this, it may be more susceptible to DNA damage, and the drugs may be more effective on these cells compared to, let's say, wild type with a fully functional Fanconi pathway. To do this, we first had to engineer the cells. We had to use a CRISPR-Cas9 system, and we engineered the guide RNA to target the BRCA2 gene. And I'll be testing, my part was using drug screens, and I'll be testing whether or not if cells with a heterozygous mutation within the BRCA2 gene are more sensitive to drugs that target these DNA repair pathways. This is just a visual of the Sanger sequencing we've done on some of our clones. And it may be hard to see, but over here, you can see that there is a heterozygous mutation. You can see peaks. After everything's, there's a friendship mutation, so everything is not aligned. It's kind of hard to see, but you do see it's not aligned as well. So this is just verification showing that our clones are heterozygous, the mutant clones. And these are the drugs I used. I use ATR, ATM, CHEK1, CDC7, CDK9 inhibitors, and they all targeted signaling kinases. A kinase is an enzyme that can phosphorylate, and it's usually for signaling. And And to do the drug screens, I first had to plate 5,000 cells per well. I then had to incubate the plates at 37 degrees Celsius and add varying concentrations from 0, which was the control, which is just media, all the way up to like 5,000 nanomolar or 10,000. We used different drug screens, but it was around 10,000 to 5,000 nanomolar, and it varied. And I assessed viability using a cell titer glow assay. Cell titer glow is a cell viability assay, and the number of cells present are quantified through the amount of ATP in a solution. So then we compared viabilities between wild type and heterozygous cells to assess whether or not how they react to the drugs, and to see how, like, the viabilities between the two. These are the cell viabilities for the ATM and ATR and CHEK1 inhibitors. In the parental and Cas9, these are both wild types, so they have a fully functional Fanconi pathway. And the bottom two are mutants, and they have an impaired Fanconi pathway. As you on the y-axis, you see a cell viability in percent, and on the x-axis you see drug concentration in nanomolars. And for these three graphs, you don't see really any of a difference in viabilities between the wild type and mutants. But you do see in the KU60019 drug that parental is a little bit more viable at 5,000 nanomolar than the heterozygous cells, but it's not by much to draw any conclusions at all. This is the PHA767491 drug, and it targets CD. It targets CDC7, CDK9 proteins, and you can see that there is increase in viability compared to the wild type and heterozygous mutants. At between 500 and 1,000 nanomolar, you see that the wild types are still 100% viable, while the heterozygous cells are around 60%. But as you can see as well, the error bars are they're still pretty large, and they're also above 100%, which could be due to a pipetting error that could be addressed. And I wanted to redo this experiment to see whether or not we see similar results and viabilities. And again, we do see that the wild type cells are still more viable than the heterozygous mutant cells when exposed to the CDC7, CDK9 inhibitor. And from 500 to 1,000 nanomolar, you still see that the wild type cells are still around 100% viable, while the heterozygous cells are lower, around 80 to 60% viable. But due to we still cannot draw any solid conclusions. We just see that there's an increase. In conclusion, viabilities between heterozygous and wild type cells are similar when exposed to the CHEK1 inhibitor, to the ATM and ATR inhibitors. But we do see that there's an increase in sensitivity to heterozygous cells when exposed to the CDC7, CDK9 inhibitor, around 500 to 1,000 nanomolar. And it'll be interesting to look into the molecular link between CDC7, CDK9 inhibitors, and the Fanconi BRCA pathway. And data for the PHA767491 drug, which was an inhibitor for the CDC7, CDK9 proteins, is most promising. But the IC50s, for the concentrations we used in our experiments, are relatively high compared to the IC50s. And IC50 is it's a measurement showing what concentration we use these drugs. And for these experiments, I use them relatively high compared to what they are and what they use in patients. So we're going to continue to expand our drug screens and concentrations to see whether or not we find any drugs that we can see a significant difference, and if we see a pattern. And we're going to also use this in a mouse model. The drugs we're also going to use, like I mentioned before, were WEE1, CHEK2, isomerase 1 and 2, hydroxy, and 5-fluorouracil. So we're going to use these drugs soon, actually. And also we're going to plan to use it in a mouse model with a heterozygous mutation in the BRCA2 gene to assess what drugs are effective in this mouse model. And for acknowledgement, I would like to thank DanaFarber, Harvard Cancer Center. I'd also like to thank the CURE program. I'd like to thank Ms. Karen Burns-White, Emily McMains, my PI, Dr. Alejandro Gutierrez, my mentor, Dr. Gail Puglia, and my other mentor, James Degar. I'd also like to thank the Biogen Foundation for the funding, and also like to thank my family for being able to support me through this and give me transportation, because it was a haul. And that is it. Thank you.